Sean Mahoney. I'm going to be your chair for this, hopefully not too long delayed session. Our first invited speaker here today is Bart Deplancke. The Deplancke lab has been made many innovations on both the wet lab side and the bioinformatics side. I'm not going to take up a huge amount of time introducing all those innovations at this point. Still not there. Well, so I have to. Okay. His lab has made wonderful contributions in understanding differentiation, especially adipogenesis. And so hopefully we'll hear lots about this in a moment. Okay, I'm going to get off here. So pressure off.
Do I get a pointer? This one? No. It's coming. What's that?
You take that?
Yes, thank you. All right. That's the one. All right. Okay, so just next. All right, okay, cool. Well, thanks everyone for staying put while you're missing maybe many other talks here at this exciting conference. So thanks so much, Sean. Thanks for the introduction. Anthony, Judith, thanks so much for the invitation. It's a pleasure to be here. It wasn't too far, but I was still excited to actually jump on the train this morning. So without further ado, I'm going to talk a little bit about genetic variation and how this influences organismal phenotypes, as the title implies. And so, as many other labs actually here, probably in the audience as well, our lab is really interested in understanding the impact of genomic variation on phenotypic diversity. And similar to many other labs, of course, we always deal with this great black box in the middle between genomic variation and phenotypic variation, which we hope, similar to others, actually to break down by simply focusing on the intermediate phenotypic traits, which in our case are the, let's see, I don't think I have a pointer. I don't think I have a pointer. Well, I will use my long arms. So the molecular traits, which we then can interpret in the context of molecular networks, and then these molecular networks can then perhaps tell us something about phenotypic diversity. Now, I'm actually going to focus on a little part in the lab that, thank you, that is involving actually Drosophila, because we still believe that basically there's great power to the model. And I will hopefully illustrate that with the type of phenotypes we're actually studying, especially since for Drosophila also, there's several population resources available that, for example, Eileen is also using, Eileen Furlong, who actually talked this morning. And just as a recap, this is the resource we're also using in the lab. This is the Drosophila Genetic Reference Panel, which has been started by Trudy McKay at North Carolina State and sampling Drosophila from local fruit markets. And then through inbreeding, she was able to generate roughly 200 of those inbred lines, which then actually provide an endless supply of individuals with the same genotype, and then you can use those actually to really do thorough phenotyping. You can look at the effects of genomic variation, of course, look at all kinds of molecular traits. All right. So today I'm going to tell you about two vignettes, two stories that have been ongoing in the lab that illustrate our broad interest actually in understanding phenotypic diversity. And the first one I'm going to talk about is actually our efforts to understand variation in gut immunocompetence. Now, it's pretty obvious why we actually would like to study this in Drosophila. It's very difficult to infect people knowingly with a pathogenic bacteria. Yet we all understand that diarrhea and enterocytic infections are still a huge problem throughout the world. There's also clear inter-individual sensitivity. Some people can eat something that some others actually will really be sensitive to. And as already illustrated or indicated, in Drosophila, we can really model this quite well, given the conserved pathways and morphological properties of the Drosophila gut compared to the human gut. All right. So this is a story that was performed in collaboration with Bruno Lemaitre's lab at EPFL, and really driven by these outstanding students, Maroon, Michael, and Danny, in collaboration with Roderick Gigo and kind help by the core facilities. And so we started this several years ago, and this was the first experiment we undertook. What we did, we took 140 or of the most vigorous lines, and we infected those with what is known to be a very pathogenic bacteria, Pseudomonas entomophila, at least for flies. So we fed those actually through sucrose feeding and then compared actually the overall survival of those flies compared to the controls. Now, what we do know about Pseudomonas is that it tends to be quite lethal, so most flies actually will succumb to this infection. And that's why we were actually so surprised to see the effect of actually population genomic variation on this particular resistance phenotype, because what we saw is the following. You can clearly see that this is truly a trait because there's continuous phenotypes all the way from flies being highly resistant, so none of those flies actually in this particular line succumbs to the infection, whereas here in this particular line, all the flies actually succumb to infection. And I think I probably can argue with you that there's probably not a more extreme phenotype like this. You're either alive or you're either dead. And so basically since then we've been really wondering in the lab where this phenotypic diversity from highly resistant to highly susceptible is actually coming from. And so we've been performing actually several experiments. I'm going to tell you about one in which we actually tried to look at molecular variation in the two extremes of this phenotype, the highly resistant ones and the highly susceptible ones. So we took about 38 of those lines. We actually then, again, infected them, isolated the gut, and then performed RNA transcriptomics, RNA-seq-based transcriptomics, to look for genes that actually might co-vary between resistant versus susceptible lines. Okay. And so, of course, you can do many things with these kind of population-based transcriptomic data sets. But one of the things we were very interested in is to first look for variants that actually might be responsible for the type of gene expression variation that we might see in this population. For those not familiar, what we're simply looking for are variants such as those here that induce a heritable change in gene expression. For example, all the individuals with this C polymorphism will actually have low levels of transcript T, and all the individuals with this polymorphism G will have high levels of this transcript T, indicating that this is what we call, therefore, an expression QTL. And so when we performed this kind of analysis using our RNA-seq-based transcriptomic data, what we saw, or what we found, is quite a few eQTLs. They're actually shown here. But interestingly, few actually were shared between the two conditions of control and infected. In fact, most of them were actually unique to a specific condition. And this is perhaps not new. It's actually now shown that there's a lot of cryptic variation out there in the genome that acts only in a particular context or at a particular time. And so regulatory variation is truly time and environment specific, has been nicely shown actually by other labs during development, during differentiation, also depending on the tissue, and also infection related. So this was not actually a surprise, but it was still very nice to see that there's so many variants actually that only appear when you actually infect flies, and these variants then, in that particular context, change the expression of many genes. Interestingly, we did not observe actually a striking difference in the distribution of these variants that affect gene expression, as is shown here. What we show here is the density of these eQTLs with respect to the transcription start site and the transcription end site of genes. And as you can see, shared, infected, or controlled, they all kind of overlap in that most eQTLs will be found around the transcription start site, and there's also enrichment around the transcription end site. All not that surprising. However, what we actually were interested in, and is something that I think we can nicely now exploit the power of Drosophila, is to understand how these particular variants act. And since these mostly are local, it's assumed that most of these act in cis, meaning that these variants actually affect only the allele located on the same chromosome. Of course, there's also another possibility in that they actually act in trans. Even though they might be close to a gene, they might still actually affect both alleles that are present. And so one has to be careful when actually calling a variant cis eQTL simply because of its distance or its near distance to a gene. This actually has to be demonstrated by its true effect on inducing allelic expression. And so this is what we actually aim to now investigate, I think, in a very kind of large-scale analysis. And here again, we exploited the DGRP because what we did is do a round-robin cross where we actually bred or crossed basically individual lines together, yielding F1s, okay? 19 of them in this case. And so for each, therefore, now we can actually look whether a specific variant of interest induces a specific allelic imbalance in terms of gene expression. Okay? So the analysis is actually shown here, how we did it. So again, we distinguish between infection and non-infection. We dissected the guts of these 19 F1 flies, and then we used actually a newly developed method in the lab, which we call BurbSeq, for an ultra-affordable high-throughput transcriptomics approach. I'm not going to go into detail, but I'd be happy to talk about it later, which has dramatically reduced the cost to perform transcriptomics to a couple of dollars or euros per library. We've then used this approach to then generate the reads. We assign them to either the paternal or maternal allele, and then we use this general linear mixed model with the response here modeled by a binomial test, since we're dealing with read counts, of maternal versus paternal reads, and then the crosses as random effect. And so the result of that is very surprising, and again, I welcome very much your input here, because what we noticed is actually that very few of what we thought would be truly cis EQTLs act actually in cis. This is illustrated here, so if you here have the number of EQTLs, we were able to test 80%, so meaning thousands of EQTLs actually for their effect on allelic imbalance. And what you see here is all those that were tested, and of those, basically only a small fraction, 20% actually, truly led to an allelic imbalance, indicating basically that roughly 80% of those local EQTLs, even though they're actually quite near the transcription start site, do not seem to actually induce an allelic imbalance, indicating that they actually act on gene expression through some other ways. I can't go into too much detail because I'd like to also talk about another story, but I'm going to quickly summarize, therefore, that the resistance to infection is truly a complex phenotype driven by variation from mostly after system perturbation, so cryptic variation. EQTLs are mostly condition-specific, but perhaps even more surprising, we seem to see that EQTLs tend to overestimate the amount of cis-acting EQTLs. In data I don't show you, but we actually have the data. It seems that neither proximity to the target gene and significance of the local association are good predictors to say whether a variant will act in cis or not. Of course, there's possible confounders to our analysis, which I'd briefly like to mention as well. There could be trans-acting EQTLs who kind of put the cis-acting EQTLs in a shadow, so they might have a stronger overall effect. There could be LD issues, linkage disequilibrium issues. And of course, a combination of variants may also induce an effect. But overall, I feel that our story is actually pretty robust to the finding that actually most variants, even though they're actually local and very near a specific gene, do not actually seem to act in cis. Going back to our transcriptomic data, ultimately we were interested to understand what the transcriptomic differences are between resistant and susceptible flies. So just as a kind of a control, we first looked at the gene expression differences between the infected and non-infected flies. And here, what we saw is already quite established. We saw a high amount of genes that actually changed their expression, either upregulated or downregulated after infection. But quite interestingly, even though basically we're talking about dead or alive, what we found actually between resistant and susceptible lines in terms of differences was actually very minimal. In fact, using a fairly conservative but nonetheless quite established approach for a twofold difference or more, we found actually that there was only one gene actually that was truly different between resistant and susceptible lines. So this doesn't happen a lot in genomic analysis, I'm sure you're familiar. So here we actually had one gene, so we only had one choice. We had to focus on Nutcracker. All right. We first established basically that indeed, if you actually knock down Nutcracker in healthy flies, so to speak, and then you infect them again, that this dramatically affects survival, either actually when maintained over a balancer. This is Drosophila genetics, but it just basically illustrates that Nutcracker mutation as a homozygous actually is embryonically lethal. But also actually in a homozygous situation, again, we see the same effect. Losing Nutcracker actually reduces your resistance to infection. Okay, so then we looked actually at the locus of Nutcracker. Interestingly, we found one variant that we actually previously had predicted to be a local EQTL. It was about 7 kb upstream of the transcription start site, and it seemed to actually cover binding sites for the transcription factor broad or daughterless, which I'm going to come back to in a moment. So that we then looked actually at what the effect was actually of these particular variants basically on the expression of Nutcracker. And as you can see, basically, here you can see that in the alternative context, Nutcracker is highly increased in the resistant versus the susceptible lines. And this actually also correlates with the fact that in the alternative allele, or for flies that actually are alternative homozygous for this particular variant, many of them actually are more resistant than in the reference context. All indicating that perhaps this variant at least contributes in a specific fashion to the resistance to infection. So we followed up on this idea that broad might be involved here, and so in order to follow this up, we actually performed an in-house protein DNA interaction assay based on microfluidics. I'm not going to go in too much detail, but basically what we do is to immobilize a transcription factor, in this case, for example, broad. We expose it to different concentrations of DNA, and this allows us then to actually plot a DNA binding curve or a dissociation curve. Okay. And we actually looked at all the possibilities for motifs that were predicted there, so the corresponding transcription factors, and only broad actually seemed to be majorly affected by the variant in this particular binding site, in that basically the reference allele actually showed very nice binding, as you can see here, but the alternative allele basically shows much reduced binding, indicating that basically in the alternative allele there's less binding by broad. Now, this is intriguing because actually it's well established in the literature of Drosophila literature that broad is typically acting as a repressor. Therefore, less binding by broad might actually lead to a higher binding or higher expression of Nutcracker. And this is exactly what we could now test because we already performed this round-robin cross actually with oral flies. And here you can actually see all the fly lines which actually had a heterozygous combination of that specific variant. So we could use those lines basically to test whether indeed this local variant is actually inducing an allelic imbalance at the Nutcracker gene. And as you can see here, if you plot the ratio of alternative reads mapping to the alternative allele over the reference allele, you can see that actually in the infected context, indeed, there's a significant skew towards the alternative allele, indicating indeed that basically in the alternative context, Nutcracker is actually increased in higher amounts. And so that leads us to the following model, where we think that basically this specific variant that actually relates to Nutcracker in the uninfected condition is basically not doing much. There's actually low expression of Nutcracker basically for both contexts. But in the infected condition, what seems to happen is that Nutcracker is induced through other regulators which we haven't identified, and then broad seems to come in, perhaps in an attempt to dampen the overall immune response, and then tries to repress basically Nutcracker. But of course, as I showed, in the alternative allele, broad will actually bind less, and therefore there will be less repression. And since Nutcracker is required for higher resistance, the higher induction of Nutcracker will lead to higher resistance at the end of the day. So this is one model in basically in which regulatory variation seems to influence actually a very important phenotype related to gut immunocompetence. All right. So switching gears, a second vignette or a second project I'm going to talk about relates to our interest in understanding basically circadian gene expression, and in our context, circadian gene expression variation. And this is work actually driven by a postdoc, great postdoc in the lab, Antonio Malares-Filho, and all the bioinformatic analyses were done by a fantastic student, Masha Litovchenko, in the lab. Why is this important? Because I think many of you will relate, maybe even this afternoon, to the fact that basically we have many circadian phenotypes. The most extreme ones are those people who actually cannot get up in the morning, or those people that actually can't get to sleep, the morning and evening people, so to speak. Okay. And this is again a phenotype that actually we can try to model quite, or that we can model quite well in Drosophila, because as you may actually remember, in 2017, the Nobel Prize was actually given for the molecular mechanisms driving the circadian clock, Hall, Rochbas, and Young, and all of this work actually was done in Drosophila, namely discovering that basically the clock was driven by a clock and cycle, which actually then induced the expression of period and timeless, and those together then again repress clock and cycle, leading to a negative feedback loop, which then induces the typical cycling that you see for circadian gene expression. And interestingly, Drosophila also shows great circadian plasticity. This is already well known actually for many years. For example, here, this is a bit of a complicated plot if you're not familiar, but what you see here is basically dark and light regimes, And so what you see is basically the black lines are the activity of Drosophila. And as you can see, right before the light goes on actually in your incubator, Drosophila will actually start to wake up, move around a little bit, and then go a little bit to a dormant state. And then it will anticipate basically the dark light or the dark context by actually waking up again, being very active, and then going to sleep again in the dark. Interestingly now, when you actually induce those flies actually to a complete dark regime, so there's no longer light and therefore no longer entrainment, what you can nicely illustrate or see here is that the circadian time actually, or the phase, becomes longer and longer, at least in this particular strain, Canton S. And in contrast, another strain actually, here the circadian cycle becomes shorter and shorter if you basically let those flies go free, right? It's a bit similar to our alarm clock actually forcing us to wake up. If you basically don't have the alarm clock, your own circadian time will induce you actually when you want to wake up, okay? But how this is actually driven is actually very poorly understood. What are the molecular mechanisms? And so this is actually what we decided to look into. We again wanted to bridge this geno to phenotype basically gap, specifically in this case by looking actually at gene expression variation over the circadian cycle, okay? So for that, what we actually did is to first actually plot a reference transcriptomic data set, which is actually performed every 2 hours for 48 hours, first in 12 hours of light, and then just to make sure that this is true circadian gene expression or circadian cycling, we then actually also sample those flies in 36 hours of darkness. We did this also in 4 different tissues, leading to 96 transcriptomes, and again, we used our own BRB or burp-seq approach for this analysis. But ideally, we'd also actually now like to incorporate genotypic information in this particular equation. And for that, we were a bit puzzled, because ideally, what you then would like to do is to look at 140 lines and do exactly the same thing here as for the white minus, which is to sample those flies every 2 hours for at least 24 hours and at least a couple of tissues. Now, if you plot that, that would lead to 5,000 transcriptomes, okay? So that's a bit too much, so we kind of scratched our head and said, how can we still actually get dynamic information perhaps, but from static sampling? And so we came up with the following approach, namely to actually sample each of the lines only once, but across the 24-hour time period, such that basically each line actually represents a specific time point in the circadian cycle, so to speak. So flies are collected every 9 minutes for 24 hours. We only have 1 DGRP line per time point, so every line will be sampled only once. And that means, therefore, that we need to actually perform or infer temporally dynamic patterns from static sampling, as we've done here. But this is actually a problem that is of great interest to the field as a whole, the biomedical field even, because often transcriptomic data sets come in from the hospital, and in order to actually understand, for example, a specific response to drug treatment, it's very interesting to know in which circadian cycle or which particular time point in the circadian cycle a patient was when this patient was actually sampled. And so several years ago, basically, there was actually an approach called molecular timetable that actually allows you to infer basically dynamic information from static sampling. But that, of course, means that we really would be able to recapitulate basically the actual cycling based on the information retrieved from 140 lines. And this is what I will show you here, that this is indeed the case. So basically, if you, for example, look at timeless expression here in the gut, you can see this very kind of striking cycling pattern for timeless. And when you now actually look at the patterns you get, basically when you sample all the 140 lines but only once, meaning that each dot here is actually one line, okay? You can again see very nicely the same kind of pattern. So this illustrates basically that our static sampling approach is able actually to produce basically the dynamic patterns that you might expect if you just base this on one particular genotype, such as white minus. I hope this is clear to everyone, because this is key for what actually remains in this talk. So again, each dot here is one line, and basically by simply sampling each line all the different lines every 9 minutes, basically you get this particular very canonical pattern of timeless cycling that is actually corresponding to when you would do this basically only on one genotype. However, as you will probably appreciate, what are these orange dots? These orange dots basically are the ones we're really interested in because these are the outliers, meaning that actually this particular line shows an expression response that actually should be here somewhere, yet you can clearly see that it's actually nowhere near the peak of expression that it actually should be, indicating that those are the lines actually that show true circadian gene expression variation. And as I already or indicated, we now can use this molecular timetable approach to systematically look as to which extent basically circadian gene expression variation is present in this Drosophila genetic reference panel. And the results basically that we got are quite striking, I think. So this is illustrated here. Here we have the 3 tissues. Each number basically is a different DGRP line, and all of those lines basically, at least in one tissue, show basically striking and significant circadian gene expression variation, indicating that basically expression in that particular tissue, for example here in line 21 in the gut, is significantly desynchronized basically with the normal circadian time that you would actually expect based on the other genotypes. In some cases, this is quite extreme. For example, here in this line, all 3 tissues actually show very aberrant and highly variable, basically, circadian gene expression. But mostly, basically, most of the variation is actually present only in one single tissue, indicating that the determinants act mostly at a tissue-specific level. But interestingly, and again, I want to reiterate that, this basically means that in flies, basically, certain tissues, for example, in this case, the gut, is completely desynchronized actually in terms of the overall circadian clock with the other tissues actually in the same fly, right? So this actually indicates that flies exhibit circadian level expression desynchrony, and this is basically at the single tissue level, which is, to me, actually quite remarkable that basically within the same organism, you can have this particular desynchrony between individual tissues, okay? So in order to understand this a little bit better, we went back to actually our reference time set for true circadian gene expression. And using this classical GTK cycle, we actually found roughly 1,729 genes that cycle in a circadian manner in Drosophila. Many of them actually only do so then in one tissue, as you can see here, for example, in the fat body, the gut, the brain. This is the kidney in the Drosophila, the Malpighian tubes, okay? Some of them are shared between tissues, and here you actually have those. Only 14 actually seem to cycle in all tissues that we examined, among which basically all the known core circadian genes also kind of validating approach. Okay. And so now what we aim to do is to understand basically how it is possible that circadian gene expression variation manifests itself at the single tissue level or in a tissue-specific fashion. And for that now we used actually our gene expression data to infer the underlying gene regulatory networks that actually drive this circadian gene expression. And for that, we use this dynamic gene tree, which is basically a machine learning method that infers regulatory information based on this time series expression data. And again, this is a complex plot, but let me please guide you through it. What you need to kind of appreciate, hopefully, is that there's 4 really main hubs here in this network, which are indeed consistent with their role actually in clock control in all tissues. These are the genes. So this is actually what you would expect. But interestingly, all of those nodes basically are now connected to smaller modules, which are highly tissue specific, and also actually are composed of cycling transcription factors themselves. So this suggests that basically there's a second level of regulation in the clock hierarchy to generate basically local gene expression rhythms. So central regulators are basically corroborating or collaborating with local regulators basically to induce circadian gene expression, but at a tissue-specific level. Interestingly, I already mentioned that these transcription factors cycle in themselves, but they do only so in that particular tissue because they're actually expressed also in the other tissues, but there they do not cycle whatsoever, indicating that there's even a higher complexity of regulation than we actually appreciate here in this plot. So coming to a conclusion here, what we've actually generated, I think, is a rich data set for circadian biology with high resolution and more than 700 transcriptomic samples. We've identified actually putative new clock genes. I've mentioned basically that we identified our core clock genes. These are 7 of them, but we identified another 7 basically that might actually also be involved in the central clock. I've shown you that there's pervasive, mostly tissue-specific circadian gene expression variation. And this variation seems to be mediated by a multi-layered circadian gene regulatory network involving central regulators, as already indicated, corroborating actually with local regulators that cycle in a tissue-specific fashion. So again, bringing all of this together, this actually might indicate that genetic variants here that drive the circadian gene expression variation may actually mostly act through these local regulators to therefore affect circadian gene expression. Okay. And so with that, I want to thank actually all the people involved, especially Michael, Vincent, Daniel, Masha, Julie of the lab, my collaborators here within DPFL and outside, and you for your attention, and I'll be happy to answer questions.
Thank you for a great talk. I just wanted to ask a clarification. So when you're talking about the genes that get desynchronized between tissues, is that based on only the constant darkness gene expression data?
Can you repeat that again?
If is that based only on the data that you collected in constant darkness?
It's basically so, yes. So in order, so that's typical for the field. So in order to make sure that you're actually dealing with circadian gene expression, you let the clock roll basically, but then in total darkness. Yeah. And so this is indeed based basically on the actual modeling of the entire cycle.
So will you expect the synchronization of the tissues to come back if you basically brought them back to the light?
I don't think so. I think this is really something intrinsic actually to the flies themselves. I think there's really a genetic component basically that acts, I think, through these local regulators that act in a tissue-specific fashion, are affected perhaps by these local variants in their binding pattern, and therefore that actually leads to tissue-specific circadian gene expression variations. So,
and just one last question. When you said brain, is that SCN or is that only brain?
This is the whole brain. This is the whole brain.
So have you looked at what is the expression only on the SCN?
Yeah, well, we've actually not looked specifically at the expression. I mean, there's now the single cell data sets and so forth, but we've actually knocked the central regulators down using, for example, the timeless drivers and the PDF driver. And actually, at least for 4 out of 7, we get actually clear circadian phenotypes. So that at least suggests that in those actually circadian neurons, there is actually some action of those particular core genes that we think, and therefore they might actually truly act in a central fashion.
Thank you.
Sure.
Judith.
Yeah, I was, so great talks. Great 2 stories. I have a question to the first one. I was really intrigued by this 20% that you said it's only 20% cis. So what about what do you think is the mechanism then? I mean, is this like the depleted of transcription factor binding sites, for example? Would you think that, because I would expect that these 20% would be transcription factors, then what's the rest?
Yeah, well, so if I may, I don't know if it's still there or not. Maybe, I think I actually prepared a little bit because I didn't have quite the time, but basically what we could do as well is that actually many of those, of the 19 lines also actually present homozygous context for those particular variants, allowing us actually to look for trans effects. And so at least we now know that, it kind of, it's not completely foolish in the sense that at least, I mean, this is a bit statistically underpowered, but for at least another 10%, we have true evidence that even though they're actually very close to the transcription start site, they act in trans, right?
So it's like non-coding RNAs or something?
Transvection?
Transvection.
I see, okay.
The enigmatic word, transvection. Yeah, exactly. I don't know.
Okay.
Yes, as always. But to me, it's very intriguing. Any input would be, because we thought we actually would we did this more as a validation of our cis-EQTLs until we found that actually only 20% could be validated as cis. So,
amazing.
Yeah. Mm-hmm.
Thanks, that was the same question.
Okay.
I find it quite shocking also, but yeah, something to discuss.
Afterwards, yes. I'd be happy to.
Thank Bart again. Thank you.
Yeah.